Hi again. This is your second and final lecture video on the unit of blood. So we've already learned about the parts of blood and what each part does and some of the disorders that go along with being out of homeostatic balance when those parts are not as they should be. So today we're going to move on to how we actually make the blood in our bone marrow. And then we're also going to learn about blood groups and blood typing and donating blood. So the word hematopoiesis was on one of your assignments, and the definition of hematopoiesis is the process of making blood cells. Hema, remember, was blood, or hemato is blood, and poiesis is formation or production. So that is the production of your blood cells. This takes place in your bone marrow, mainly in the flat bones that are found in your skull and your hips and your sternum and rib cage, which is known as your thorax. And it's also in the long bones of your arms and legs, like your humerus and your femur bones. So you have a hollow cavity in the middle where the bone marrow is located. And inside that bone marrow are your stem cells called hemocytoblasts. And so remember, we've already broken that word down. It means blood cell generative or germ bud. So those are the cells that have the ability to form into any of the three types of formed elements. And that would be found specifically in your red bone marrow. The rate of production of your red blood cells is controlled by a hormone called erythropoietin, and the kidneys produce this hormone. So it doesn't really matter what your number of red blood cells is. It's more important how well are those red blood cells transporting oxygen. So that's what determines the rate of production of your red cells, is whether or not they are efficiently transporting oxygen. So if they're not, even if your red cell count is normal or high already, you're still going to produce more red cells because you're having a hard time transporting oxygen. Your red blood cells only live for about 120 days before they start to fragment and break apart, and then they have to kind of be eaten and broken down by the phagocytes so that your body is kind of able to reabsorb those broken down red cells, and then you're able to replace them with new young ones. And they take about 3 to 5 days to mature, and during that time you've got the organelles, you've got the nucleus, and the red cell is producing all this hemoglobin, and then after about 5 days it spits out all of its organelles, and it's including its nucleus, and it simply keeps the hemoglobin in there. So basically your red cells are like bags of hemoglobin that are then able to go around and attach oxygen to them. In terms of forming your white blood cells and your platelets, you have what are called interleukins for producing white blood cells. And they're either going to prompt the white blood cells to be produced in the red bone marrow, or they can also use this hormone called interleukin. And there's many different kinds, and where it's too complicated for us to learn about all the kinds, so we're just going to group it to one category called interleukins. We can also use different interleukins to assemble the army or form up the troops so that we can fight off infection when we're being attacked. And then the hormone thrombopoietin is what would involve the production of platelets. So everything is determined by your hormones getting released from your glands. And of course, remember, your brain would be the control center that's sending the message to the effector organ of the gland to produce that hormone, and then you would get the desired cause, or excuse me, the desired effect of producing more of that type of blood cell. Hemostasis is the scientific name for the stoppage of bleeding or for blood clotting, because hemo is blood, stasis is standing still. So when your blood is standing still, that's kind of like what happens when you form a blood clot. There are 3 major phases. The first phase is called a vascular spasm. And what happens is when a blood vessel is injured, the blood vessel immediately constricts so that you won't lose too much blood. And this causes the vessel to spasm. And then that in turn causes the second stage, which is the platelets become sticky and they cling to the damaged site. And then eventually that leads to the third stage, which is where coagulation, or the clotting, actual clotting of the blood forms. And it works with, there's lots of very complex terms, but we're going to try to keep it simple and just say there's this protein called fibrin that begins to form this mesh-like trap for the red blood cells so that you can actually form the clot. And then that's what you see if you have ever looked at a scab up really close. It basically does look like mesh, and that's all that fibrin that has trapped the blood. It takes about 3 to 6 minutes for this to happen. And then once the clotting has started, you actually want to trigger the inactivation of all those factors, because you don't want the clotting to become widespread and then you're having trouble getting your blood to flow through your body. Some disorders of hemostasis. One is called a thrombus, and a thrombus is a clot that develops in an intact blood vessel. So there hasn't been any damage to the vessel, and a clot forms anyway. And that can cause problems if the clot's large enough that it causes circulation issues. And sometimes that thrombus, that clot, can even detach itself from the vessel wall, and then it's kind of flowing through the bloodstream itself, and that's called an embolus. And of course, if it gets into a vessel that's too small, then it can cause blockage for the circulation, so that can be a problem. Anytime the vessel wall, the inside of it called the endothelium, anytime it gets roughened up, which could be from getting burned, some kind of physical blow, or the accumulation of fatty materials inside your vessel, that's going to cause undesirable clotting. And then you got all these platelets that are clinging to each other, and then that can cause problems. Thrombocytopenia is a condition that involves an insufficient number of platelets. Okay, so the movement that you do in your body, just the movement alone can cause bleeding from the small vessels, and this can cause you to have these purplish blotches on your skin, and that's known as petechia. And this can be caused by anything that's going to suppress your bone marrow. So when you have an insufficient number of platelets being made by your bone marrow, then you can suffer from petechia and thrombocytopenia. Hemophilia is another disorder of hemostasis, and that's where it's any kind of blood disorder that's hereditary that results from a lack of clotting factors. So then you have trouble clotting your blood. So some hemophiliacs do not have the ability to clot their blood at all, and they have to be rushed to the hospital and given a shot that forces their blood to clot. So it can be a very dangerous disorder. All right, the last thing we want to talk about in this unit on blood is human blood groups. And so we're going to learn about different blood types for humans and how you determine someone's blood type. So your blood type is going to be determined by the protein markers, or what we call antigens, that are on your red blood cells. An antigen is any foreign substance that the body can recognize. However, on your own red blood cells, those are antigens that your body can recognize as self-antigens, so they're not seen as foreigners. And so you can contain one of 3 different types, or numerous. You can have more than one of these different types of antigens in your blood. And so with the 3 antigens together, you can have 8 different possible combinations of those antigens to determine the 8 different blood types for humans. So the 3 antigens are known as the A antigen, the B antigen, and the Rh antigen. And your body is going to form antibodies when exposed to any foreign antigens. Okay, and those bodies are going to attack those foreign antigens, which is going to cause agglutination or clotting in your blood. And this, of course, can cause death for you. So you really want to make sure that you're getting blood that is compatible with yours, or you're going to attack that foreign blood, which is going to end up harming you as well. When you type someone's blood, you're going to mix their blood with those 3 different types of possible antigens, although you're going to mix them with the antibodies for those antigens. And so they call them anti-sera. And so there's an anti-A serum, an anti-B serum, and an anti-Rh serum. And so what you'll do is you'll have 3 different wells on the slide, and you'll put a drop of blood in each well, and then each well gets its own sera added to it, its own serum. So one of them you'll add the anti-A to, one of them you'll add the anti-B to, and one of them you'll add the anti-Rh. And you're looking for clotting, or what's called agglutination. So if agglutination occurs in the anti-A well, then that must mean that your blood has the A antigen, and that allowed the anti-A serum, or what would be known as antibody A serum, to attack your antigen. If no clotting occurs, then it means that you don't possess that antigen. So we can use the wells and looking to see if there's clotting or not to determine which types of antigens you have in your blood. When you look at the U.S., this is a breakdown of the different cultural makeups. We have white, black, Asian, and there's Native American. There's lots of other cultures in the United States as well. But this just gives you a general breakdown of the different blood groups. It doesn't include the positive and the negative, so I'll give you some data in class that goes further into just in general for the United States, what percentage of individuals in the U.S. have type AB negative versus what have type AB positive, and so on. So I'll give you more information about that. But this is a great chart to refer back to so you know which blood groups have, which blood types have which type of antigens on their red blood cells, and then which kind of antibodies that blood type would form, and then who can that person receive blood to. And we're also going to be learning who can you donate blood to. This is a blood chart that we'll be going through. It tells you every one of the 8 blood types. It tells you which antigens they have present and the antibodies that are formed by that blood type. So this is a chart that you'll want to become very familiar with, and we'll do some assignments in class with it. The universal recipient and the universal donor are 2 of the blood types out of the 8. And the universal recipient is AB positive. And the reason they are the universal recipient is because they have all 3 of the possible antigens. They have the A, the B, and the RH. And so they can receive blood from anyone because they're not going to form any antibodies. O negative, on the other hand, doesn't have any antigens at all. So it's known as the universal donor because it can donate to anyone because there are no antigens on the red blood cells to be able to form antibodies against. The last thing we're talking about was your blood genotypes and phenotypes, and then how you can use those to do Punnett squares that you learned how to do in Biology 1. So a couple of vocabulary terms that come back into play are phenotype and genotype. Remember, your phenotype is the physical expression of your trait, and so you would say, I have type A positive, I have type O negative. That's your phenotype. The genotype that you have represents the pair of alleles, okay, or the pair of genes that you got from your parents that code for you to have that blood type. And so the 2 come together to make up your phenotype. So there are the possible genotypes for your blood. If you were type A, you could be homozygous or heterozygous type A. So if you're homozygous type A, that means both of your alleles have the A allele on there, so you code for making the A antigen. If you're heterozygous, then one of them codes for you to make the A antigen, and the other one would code for you to not make any antigens. So you do make the A antigen. Still, you just only have 1 copy of a gene that codes for that. Same for type B. You can be homozygous or heterozygous type B. For type AB, it means one of your parents gave you the gene that codes for you to make the A allele, and one of you, or the A antigen, one of them gave you the allele to code for the B antigen, so you make both A and B antigens. And then for type O, you have 2 genes that code for you to not make any of the antigens, so you don't make the A or the B antigen. We'll talk more about the RH antigen in class, and we won't be using it when we do the Punnett squares. So then when you look at the Punnett squares, if you were going to do a Punnett square here, let's do a practice one. Let's say you're to cross a heterozygous type A with a heterozygous type B. Well, hetero means different. So heterozygous type A, they would have 1 allele that codes for the type A, so they would make the A antigen, and the other one is just coding for not making any antigens. For heterozygous type B, you would have this. So then when you fill out your Punnett square, this person would have AB, this person would just have the B, this person would have the A, and this one would make neither antigen, and that's what forces you to be type O. So you can go through and practice more Punnett squares on your own, and that's it for your notes for the blood unit.